Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I am Mokar Ahmed and this is the lecture number 2 in genetics. In the first lecture, we have studied about the basic terminology. Today we will study about Mendel and his work. Mendel, he is known as the father of genetics. Mendel is known as the father of genetics or the pioneer of classical genetics. He is the pioneer of classical genetics. Because Mendel was the first who worked on the genetics of pea plants. For the first time, who worked on pea plants genetics. And the science of genetics actually it started in 1900th century. The science of genetics, it was actually started in 1900th century. If we see that the duration of Mendel was 1822 to 1884. The Mendel worked on pea plants, but the Mendel's work it made no impression. It made no impression in his time. The people of that time, they did not accept the work of Mendel. There were several reasons that why the people not accepted the Mendel work. The reasons were that the biologists were they were preoccupied. The biologists were already they were preoccupied during his time. From Darwin's theory. With Darwin's theory. The other reason was that the Mendel published some of his work in 1866. In 1866, the Mendel published his work on experiments on plants hybridization. The topic was experiments on plant hybridization. Experiments on plant hybridization. But when the Mendel published this work, so this work was published in a journal. Journal is a magazine in which the research papers are published. In a journal, and this journal was not a recognized journal. This was the second reason. The third reason was that the people of that time, they were not familiar with statistics. The Mendelian work, it was actually confirmed in 1900. The Mendel work, I will not go to the history in detail, but shortly I will discuss only in short that Mendel's work in 1900, it was confirmed by some other scientists. And the laws that we study that the Mendel has presented 3 laws. So these laws were also formulated later on in 1900 century. Now the Mendel, he selected pea plants for experiments. The Mendel selected pea plants for his experiments. for experiments. So why the Mendel he selected the pea plant for experiments? So there were several reasons. The reasons were that it has a short life span, or we can say the short life cycle. The second one, that the pea plant it contain clear cut variations, contrast characters. It contain contrast characters. The other one is it is easily grow. It can be grow easily. The other reason was that it has the self-pollinating ability. Self-pollinating, as well as it can be cross-pollinated. It can be cross-pollinated. The pea plant it contain contrast heritable characters. Contain contrast heritable characters. Contrast heritable characters and large number of offsprings. The pea plant it produce large number of offspring, and there were also convenience in handling of pea plants. Convenience in handling of pea plants. So these were the reasons for which the Mendel selected pea plants. Now Mendel first of all he performed monohybrid cross. Mendel performed first monohybrid cross. Now what is monohybrid cross? Monohybrid cross, this is a cross between two organisms. The cross between two organisms which are different only in a single pair of contrasting character. Pair of contrast character. For example, of contrasting character. So this is monohybrid cross. So the Mendel first performed monohybrid cross. He selected two pea plants. The Mendel he selected two pea plants. The one pea plant was tall. This is tall, and the other was dwarf. So these two pea plants were different from each other only in a single contrast character. This is only one single contrast character, and that single contrast character is only the height of the pea plant, that the one pea plant was tall and the other was dwarf. Now these are the parents, and the F1 was when these both were crossed. So in the F1 generation the Mendel got TT. All the plants were tall. The F stand for it has been derived feline. Feline is a Latin word. Feline means sun. So this is sun generation. So the F1 generation it is formed by the cross of parental generations. So when the Mendel crossed the pea plants, these pea plants were different only in a single contrast character. That was the height of the plant. So the Mendel got in F1 all the plants, and all the pea plants were also tall. Then the Mendel again he crossed the F1 with F1 to get F2 generation. It is equal to F2 generation. So in the F2 generation the TT cross capital T small t. Again the Mendel got this is the F2 generation. In F2 generation, the Mendel got the phenotypic ratio was 3:1, that the three were tall and only the one was dwarf. So the Mendel, he thought that why the dwarf character, it has been conceived in the F1 generation, and why this character reappear again in the F2 generation. Then the Mendel also crossed the F2 generations, the self-pollination again. How? That this is the dwarf plant, the TT. So the Mendel crossed the F2 with F2. In the F2, the TT were crossed with TT. So all the plants were dwarf. Then this TT, the Mendel also self-pollination, also done the self-pollination. The TT cross TT. So it is clear that all the plants will be tall. So in the result, he gets F3 generation. This is now called F3 generation. Now the Mendel also crossed the TT cross TT. Now these are the heterozygous condition. So the Mendel got the two types of plants again, the tall plus dwarf in F3 generation. In F3 generation. So then Mendel thought again and again that why in the F1 generation there is no dwarf character. In F1 generation there is no dwarf character. In F2 generation the Mendel got again the dwarf character, this one. Now in the F3 generation the Mendel got the tall, the dwarf character again. Also the tall character and also the tall plus the dwarf character. So after this the Mendel presented the phenomena of dominance and recessio, and now we called it the law of dominance. So what the Mendel described in the law of dominance? In the law of dominance, this is the first law of Mendel, that we call the law of dominance. According to this law, that in the crossing between, according to this law, in crossing between two pure homozygous organisms, two homozygous pure organisms for contrasting characters, for contrasting characters of a pair, for contrasting character of a pair. So only one character of the pair, only one character of a pair appear in the first filial generation, in the F1 generation. Now this is the law of dominance. In the law of dominance, it is very clear that in crossing between two pure homozygous organisms, now these two are the pure homozygous organism, the tall. This is the pure homozygous dominant. This is the pure homozygous recessio. The cross between the two homozygous pure organism for contrasting character. The contrast character is that one is tall and the other is dwarf. Only one character of a pair appear in the first filial generation. Now out of these, only one character appears in the first generation, and this is the tall. Now in this the character which appear in the first generation, this character is called dominance character. Now the tall is the tall. It appear in the first generation, appear in F1. The F1 was T and T. So this T is dominance character. This is dominance character. And this small t, this is recessio character. Recessio character. Now, how the dominancy and the recessive character occur. Why one gene express itself and why the other gene do not express itself. It has been described by many scientists that why the one gene become or dominant and the other gene become recessive. For example, in this case, in this heterozygous condition, the allele T, it is able to express its phenotype. It is able to express its phenotype. So in this condition, this phenomenon is also called haplosufficiency. It is also called haplosufficiency. Now what is haplosufficiency? Haplosufficiency is a phenomena in which a gene in haploid form is sufficient to express its phenotype. So this is haplosufficiency. For example, the protein which is required for tallness. The protein that is needed for tallness. So this protein, the amount of this protein, for example, the amount of protein needed for tallness is 50 microgram. So it will cause the tallness of the pea plant. So in homozygous dominant condition, the T and T. So this T is also able to produce 50 microgram. It also produce 50 microgram. Now in this condition, this plant will be tall. Now in heterozygous condition, the T and T, the requirement is 50 microgram for tallness. Now in heterozygous condition, this T also produce 50 microgram. So the 50 microgram, it is enough for tallness. For this character, the 50 microgram is enough. So this is the dominant. Now this one is the dominant and this one is the recessive. Why now the T is recessive? Actually, the recessive gene occur due to mutation. The T, it will be recessive. So how this T is recessive? T is recessive mean that the T, it do not produce the protein. It do not produce the protein which is needed for tallness. So this T is called now the recessive. The other phenomena, this is the haplosufficiency. Now sometime haploinsufficiency also occur. Haploinsufficiency. Now what is haploinsufficiency? Haploinsufficiency is that when a normal gene in haploid form, is not able to express its normal phenotype, to express itself. For example, the example is that there is one of the gene and the name of this gene is CD2AP, a gene that is CD2AP. Now this gene, for example, if you represent this gene on C, now this gene, it is responsible for normal kidney function. When there is homozygous dominant condition C and C, so in this condition there will be normal kidney function. Now when there is the heterozygous condition is that when there is the normal C and the other is recessive C and the other is recessive, that is small c. Now in this condition the capital C and the small c, there will be no normal function of kidney. No normal function. So this condition is now called haploinsufficiency. That this C in haploid form, this is haploid and this is haploid, so combinedly it formed diploid. Now in this haploid form, the C is not able for the normal function. This C also need its another partner, the capital C. So this is called haploinsufficiency. The other phenomena for the dominant and recessive was also described by Japanese scientists. A group of scientists in 2010. In 2010, the Japanese scientists they described that the dominant gene, for example, there are two genes, the dominant and recessive. This one is the dominant gene. That is the, for example, if you say this is the gene A, this is the dominant gene, and the other is the, this is the small a, that is the recessive gene. So that described that the dominant gene, it produces small RNAs. This gene, it produces small RNAs. These are now the small RNAs. So that described that these small RNAs, these are actually the dominance modifier. They have called it dominance modifier. Now what is the function of the dominance modifier? That these small RNAs, it have receptor site on the recessive allele. For example, these are the receptor site for these RNAs. I will make it on another color. So these are the receptor for, so these are the receptor for that small, those small RNAs which are produced by the dominant gene. So these RNAs come and it binds to the recessive gene. So when these RNAs, when it binds to the recessive gene, so it protects the normal function of this allele of this gene. So that's why this gene will not able to function normally. Not able to function normally. So this is also one of the phenomena. But this phenomena is not possible in all genes because not all the gene contain the receptor sites for dominance modifiers. These dominance modifiers are actually the small RNAs which are produced by the dominant gene, and these RNAs bind to the recessive allele that prevent the normal activity of this gene. So this is also one of the phenomena. So this was the first law of dominance, and actually the laws were formulated later on in 1900th century, but these phenomena were described by Mendel, so that's why the scientists name it the laws of Mendel's. So this is about today's lecture. Thanks. Stay with me for the upcoming lectures.